Welcome to the deep dive. Today, we are attempting to chart what is probably the most intricate geography known to science, the human central nervous system.
It's an incredible challenge. We're talking about, what, 100 billion neurons?
100 billion.
Yeah.
And they form more synaptic connections than there are stars and planets known in the observable universe. It's complexity incarnate.
Absolutely. And trying to learn it can feel, like trying to drink from a fire hose.
It really can.
But the goal of learning neuroanatomy isn't just about memorization. It's about building an anchor, a framework for interpreting what goes wrong.
And for that anchor, our source material today is really the gold standard, the Brain Atlas, a visual guide to the human central nervous system, 4th edition.
By Woolsey, Hanaway, and Godot. It's a phenomenal resource.
And our mission for this deep dive is tailored specifically for you, the medical learner. So whether you're a student, a young professional, or you're just intensely self-taught, we're going to provide a high-yield structured summary of this text.
We're really focusing on how that structural organization connects directly to, say, the vasculature and those critical functional pathways.
Exactly, because at the end of the day, when you look at a patient with a stroke, or you're researching dementia, or just trying to interpret an MRI, you need that precise structural map.
It's the difference between knowing a brain area is damaged and knowing the exact tract that was compromised. So where should we start? The fundamental hardware.
Yeah, let's start at the cellular level. We're talking about two main populations of cells, right? The active communicators and then the essential support framework.
Correct. The communicators, of course, are the neurons. These are the specialized cells designed to convey and process information.
You can think of them structurally, I suppose. They get input from the dendrites.
Right. They integrate that signal in the cell body and then transmit the output down a single axon to the next cell. That connection is the synapse.
And their partners, the glial cells, they're so often overlooked, but they perform these absolute
vital functions. I mean, especially when you talk about forming the crucial structure we call myelin.
Myelin's the key to efficiency, isn't it?
It is. Glial cells wrap axons in this fatty, insulating sheath. And what that wrapping does is, well, it's brilliant. It does two main things. First, it massively speeds up the conduction of electrical impulses.
Second.
Because of that speed, we can pack axons much, much more densely together. And this distinction is actually how we define the basic look of the CNS, gray matter versus white matter.
So gray matter is where you find the neuron cell bodies and the synapses.
Exactly. And the white matter is just dominated by those myelinated axons and their support glia.
Okay, so we've got the cellular architecture. Let's zoom out to the gross organization. The whole adult structure is basically dictated by how the embryonic neural tube expands, right? Ontogenesis.
That's the one.
Yeah.
And understanding these divisions is the basis of all regional neuroanatomy. So if we start at the top, or rostrally, the most complex division is the telencephalon.
Which gives rise to the entire cerebral cortex, the basal ganglia, all the high-level stuff.
All the high-level stuff. Then just below that, you have the diencephalon, which contains the critical switching stations.
The thalamus and the hypothalamus.
Precisely. And from there, we start moving down into the brainstem structure.
Okay, so what's next?
That starts with the mesencephalon, which becomes the midbrain. Below that, the metencephalon develops into the pons and, of course, the cerebellum.
And finally, at the very bottom,
the myelencephalon. That forms the medulla oblongata, which transitions directly into the spinal cord. It's a developmental sequence that really dictates the hierarchy of function.
From high-level cognition in the telencephalon all the way down to vital reflexes in the myelencephalon.
You've got it.
That structural development also creates the internal plumbing system, right? The ventricles filled with cerebrospinal fluid, CSF. And the fluid dynamics here are just, they're fascinating. It's not just a passive shock absorber.
No, not at all. It's an active factory and a circulatory system. At any given moment, you have about 150 cubic centimeters of CSF surrounding everything, acting as a cushion and a waste removal pathway.
But the turnover is the amazing part.
It is. The choroid plexuses, these specialized structures, actively generate about 700 cc's of fresh CSF every single day.
700. So you're getting what, 4 or 5 complete fresh fluid changes in a 24-hour period?
Exactly. And that continuous circulation from the ventricles out into the subarachnoid space and then eventually getting reabsorbed into the venous system, that's critical for keeping the CNS environment stable.
Now, before we move on for the basics, we have to talk about terminology. There's this one conceptual hurdle every learner faces the directional terms.
Ah, yes, the biped problem. Because humans stand upright, unlike, say, a rat, we have a 90-degree bend where the forebrain meets the brainstem.
Right, so a turn like dorsal means towards the back in the spinal cord, but because of that 90-degree turn in the forebrain, it means towards the top surface.
It forces you to be very precise. It's why that precision is so necessary for reading any medical imaging, which I think leads us perfectly into our next topic.
Visualization.
Right. The real power of an atlas like this is how it connects that gross anatomy to modern clinical imaging.
And to really get that three-dimensional feel for the brain's internal structures, you have to be comfortable looking at it in the standard planes: coronal, sagittal, and axial.
And the key to linking all of those views together is standardization.
The ACPC line.
The ACPC line, that line connecting the anterior and posterior commissures, it gives us a reliable reference point for mapping structures, whether you're looking at a physical slice or an MRI scan.
So let's talk about those MRI images, because you have to know what you're seeing. T1 versus T2. What's the conceptual difference for a learner?
Okay, think of it this way. T1 weighted images are excellent for structural detail. They make the white matter look brighter than the gray matter, and crucially, the CSF looks dark.
So that's the one that most closely resembles a preserved anatomical slice. You use T1 to find things.
Exactly. Now, T2 is fantastic for pathology. It highlights areas with excess water or edema. Here, the CSF is bright, high signal. The ventricles just pop out at you.
And the brain tissue itself.
It flips the script. White matter is dark, and gray matter is often a bit lighter. So if you see a bright spot on a T2 image, you immediately have to ask, what's causing that increase in fluid? Is it inflammation, a lesion, an injury?
Beyond just structure, we have to grasp the vasculature. If you miss a vascular territory, you miss the patient's likely functional problems.
100%. And the atlas uses angiography to show this, but now we have things like MRA, magnetic resonance angiography.
Which doesn't always require an invasive procedure.
Right. MRA is brilliant because it emphasizes the signal from moving blood molecules. It actively suppresses the signal from the stationary brain tissue around it.
So you're basically just mapping where the blood is flowing, which gives you these incredible non-invasive views of the major arteries, anterior, middle, posterior, cerebral, and the deep veins.
Precisely. And that leads to what I think are the most clinically relevant maps in the whole atlas, the vascular territory map.
Yeah, these are crucial.
You look at the middle cerebral artery territory, you see it supplies the whole lateral convexity. So if a patient has a stroke there, you know immediately which functions, language, motor control for the face and arm, are at risk. Understanding these maps is the cornerstone of interpreting stroke damage.
All right, let's shift from those large structures to the organized bits of gray matter. Starting in the spinal cord and moving up, what's the basic organizing principle here?
It all goes back to an embryonic divider, the sulcus limitans. This little groove divides the gray matter into a basal plate, which is anterior, and an alar plate, which is posterior.
And that division dictates function. Basal is motor, alar is sensory.
That's the rule. So in a spinal cord, you see it clearly. The basal plate gives you the anterior horn motor nuclei for voluntary movement. We call that somatic motor, or SM, and the lateral horn for autonomic functions, which is visceral motor, VM.
And the LR plate gives rise to the posterior horn, handling all the incoming sensory information from the body and the organs.
Right. Somatic sensory and visceral sensory, SS and VS.
Okay, so when we get to the brainstem, that same pattern is there, but it gets more complex because of the cranial nerves. We have to add 2 more columns of nuclei?
We do. We add the branchiomotor nuclei, or BM. These are motor neurons specifically for muscles that come from the embryonic branchial arches.
Like the muscles for chewing from cranial nerve V and facial expression from CNCS.
You've got it. And because they're motor, they stick with the basal plate derivatives.
And what's the other new one?
That would be the special sensory nuclei, SPS. These are for unique sensory functions like hearing, balance, and taste, governed by nerves like cranial nerve 8. So the brainstem keeps the 4 basic columns and just expands to 6.
So knowing that pattern, you can pretty much predict where any cranial nerve nucleus will be and what it does.
It gives you an immediate organizational advantage.
This organization is only really valuable if we can trace the information flow. So let's compare 2 really important sensory pathways: fine touch versus pain.
Great comparison. They start in the same places, but their routes are radically different.
So let's start with touch and position sense, the posterior column medial lemniscus system.
Right. So the first order axon, say from a receptor in your finger, travels all the way up the spinal cord on the same side, ipsilaterally in the posterior columns. It doesn't cross over until it hits the medulla.
A system designed for precision, crossing high up in the brainstem. And where does it go from there?
After it crosses in the medulla, the second-order neuron forms the medial lemniscus. It projects up to the VPL and VPM nuclei of the thalamus. And from there, third-order axons shoot up through the internal capsule to the somatosensory cortex.
Now contrast that very precise high crossing route with the pain pathway, the spinothalamic system.
The pain pathway is all about speed and awareness, not high-fidelity location. So when that first-order axon enters the dorsal horn, it synapses immediately. And the second-order axon crosses the midline right there in the spinal cord.
So it crosses immediately and then shoots up on the opposite side.
Exactly. It also targets the VPL-VPM of the thalamus, which helps you localize the pain. But pain, it's different. It engages your whole being.
And the pathway reflects that.
It does. The spinothalamic tract also sends signals to places like the insular and cingulate cortices. This parallel processing explains why pain has such a huge emotional and motivational component. It hits your emotion centers in a way that simple touch doesn't need to.
Let's flip to motor output, the corticospinal tract, the system for fine, voluntary movement. Where did that command pathway start?
It starts broadly, not just from the classic motor strip in the precentral gyrus, but also from premotor areas and even from sensory areas that provide feedback. And all these descending axons funnel together into one of the most clinically sensitive spots in the brain.
The posterior limb of the internal capsule.
The very one. They pass through there, down through the midbrain and the pons, but the main event happens lower down in the caudal medulla.
The famous pyramidal decussation.
That's it. Somewhere between 65 and 90% of those fibers cross the midline there. They form the lateral corticospinal tract, which then controls the opposite or contralateral side of the body.
Which explains that fundamental rule of neurology. A lesion high up in one hemisphere causes weakness on the opposite side.
Precisely.
Lastly, let's touch on the hypothalamus, the tiny master regulator of homeostasis, sleep, feeding, all of it. How does it manage wakefulness?
It's a beautiful system. Arousal is largely driven by a specific group of cells, the orexin cells in the lateral hypothalamic area.
So they promote being awake.
They do, but they are kept in check by their counterpart, the ventrolateral preoptic nucleus, or VLPO. When the VLPO is active, it promotes NREM sleep by inhibiting those arousal centers.
So it's basically an on-off switch for consciousness.
A beautifully simple one.
Yeah.
And since the hypothalamus needs to monitor what's going on in your blood hormones, glucose, it uses these unique structures called circumventricular organs.
CVOs. And what makes them so special?
They are unique because they are midline structures that lack a functional blood-brain barrier. And that's on purpose.
So their neurons can directly sample the blood.
Yes. The area postrema can check for toxins that might make you vomit. The median eminence monitors hormones. It's how the CNS can respond instantly to changes in your body's chemistry.
We've covered a lot of ground from the neuron all the way up to these complex functional pathways, using the Brain Atlas as our guide. The real value for you, the learner, is moving past just listing facts and toward understanding the three-dimensional logic of the CNS.
And if you just think about that comparison we made between the touch and pain pathways, it leaves you with a fundamental question. Why did evolution decide that the high-fidelity touch pathway should cross so high up in the brainstem, while the immediate, urgent pain pathway crosses right away in the spinal cord?
It's a design question, really.
It is. It suggests that for threatening stimuli, speed, and a comprehensive emotional response trump precise localization, it's about survival.
A profound concept to think about. Now, to help you consolidate what we've talked about today, here is your required review exercise.
Okay. A stroke affects the posterior limb of the internal capsule on the left side. Based on the major pathways we discussed, what two distinct sensory modalities would you expect to be impaired on the right side of the patient's body? And through which specific tracts or systems are these signals traveling at that level?
And the answer is that you would expect impairment on the right side, affecting both fine touch position and pain temperature. The key insight here is that the third-order neurons for both the posterior column system and the spinothalamic system are traveling together through that posterior limb of the internal capsule to get to the cortex.
A single lesion there knocks out both contralaterally. It's the convergence point for all of the body's major ascending sensory information. Understanding that bottleneck is absolutely vital.
That wraps up another deep dive into the core material. We hope this gave you the high-yield summary you needed to master the magnificent complexity of the central nervous system.